It's things that get brought up during medical diagnosing. It's part of the conversations we have with our older parents. And what is that? Neurodegeneration. What is neurodegeneration? And what does it have to do with pathophysiology? Well, let's talk about that.
Warning. The following presentation contains information that might contradict what you have previously heard, or believed to be true, about how the human body works, and contains material that is not suitable for closed-minded individuals. Enjoy!
When we start looking at the way in which the nervous system functions, the way in which the neurons within the nervous system functions, what we're doing is we're developing distinct networks that are going to allow for passage of information that's going to allow us to be able to maintain our homeostasis, be able to remember things that we did just today, just yesterday, a week ago, a month ago, years ago, all based off of chemical messengers being sent from one neuron to another neuron at what's referred to as the synapse, or be able to send action potentials from point A to point B within the nervous system. So we're able to do what we need to do in order to be able to survive and remember what we're able to do. Part of this pathway starts during development when we start to actually grow new neurons. This growth of new neurons will continue into adulthood, just the rate at which we grow new neurons is much less than the rate at which we lose neurons throughout our life. That leads to some of the misconceptions about having less neurons than what we had before, as well as the inability to grow new neurons throughout our life. What we have to remember is that neurons themselves are amitotic. That means that they do not divide. They will branch, they will make new synapses, but they do not make new neurons from existing neurons. And what we want is we want to grow new synapses. The rate at which the neurons are being used is going to lead to stronger neurons, bigger neurons, and more synapses within the neurons. This is what takes place during the learning process. Also that's taking place within the learning process is an indication what's referred to as Hebbian synapsing. That is where the neurons that are firing together survive together, whereas neurons that do not fire together get lost in a process known as synaptic pruning. The actions of synaptogenesis, making new neurons, and synaptic pruning, the removal of synapses, will occur throughout our lifespan. Some of this leads to our ability to learn, but also leads to the ability to become forgetful. Within this Hebbian synapsing process, within this idea about making this synchronicity of neuron firing, we end up getting coordination of neurons within pathways. This allows us to remember things. However, when we stop using those pathways more often, we start to become forgetful. This is different than what we see in terms of neurodegenerative dementia. This is where it simply takes a little bit longer to remember what we need to remember. Some of this synchronicity or this asynchronicity that will occur during normal life can take place when we happen to be under high stress, such as what happens when we become forgetful when we're taking a test. But it's different than what we see in terms of neurodegeneration. When we start looking at neurodegeneration, what we're talking about distinct things happening within the physiology or within the anatomy of the neurons. One of the culprits that comes into play is issues at the mitochondria, where we do not get the correct amount of ATP or enough ATP back in order to allow for the neuron to function correctly. Another thing that might take place is growth of proteins that can become misfolded and will interfere with the ability for the axons to do what the axons need to do. We can have interferences with the ability for the neurotransmitters, the chemicals, to go from neuron A to neuron B that can lead to issues with communication between neurons that can lead to neurodegenerative issues. Or we can have issues with the support cells, in particular the myelin cells, that can make it very difficult to send very fast action potentials through what's referred to as saltatory conduction, where instead of having very short segments of the axon needing to do its repolarization and depolarization to allow for rapid movement, what ends up happening is we end up losing that myelin, and we lose that myelin, instead of having this saltatory, very fast conduction, we now have a continuous, very slow conduction taking place, which reduces the rate at which action potentials will be able to go through and allow us to have distinct types of signals. So we have different types of neurodegenerative diseases. We have inflammatory neurodegenerative diseases. We have neural death neurodegenerative diseases. We have protein aggregation neurodegenerative diseases. We have synaptic and neural network deficits leading to neurodegenerative diseases. We have different types of neurofilament diseases. We have abnormal mitochondrial functions, and then we have genetic issues that come into play. All of these is going to be different than that traditional or normal forgetfulness that will take place based off of the asynchronicity of firing within the neural networks. This is where we actually have issues taking place on the neurons themselves or in the neurons themselves. We start looking at where do these issues come into play in terms of where on the neurons are we going to see these distinct types of neurodegenerative diseases. We will have distinct types of neurodegenerative diseases that can affect the myelin cells. We have distinct types of neurodegenerative diseases that are going to affect the microglia. We have distinct types of neurodegenerative diseases that are going to affect the astrocytes. And we have distinct types of neurodegenerative diseases that are going to impact the ability for the neurons themselves to maintain their synapses. Within each types of these neurodegenerative diseases, we're going to see distinct types of functional deficits taking place. When we start looking at the neurodegenerative diseases, we have them branched out into where do we find these functional losses. We can have central diseases, diseases that are going to impact our cerebral cortex, our limbic system, or in the brainstem. We have spinal cord injuries. We have peripheral injuries. Things that are going to impact within the central is going to be things like Parkinson's disease, Huntington's disease, Alzheimer's disease, frontotemporal degeneration, multiple sclerosis, spinal cerebellar ataxia. We can have spinal cord injuries, things that are going to be like amyotrophic lateral sclerosis, ALS, or Lou Gehrig's disease. We can have peripheral diseases that can come into play. All of these various different types of neurodegenerative diseases have a host of various issues that come into play. What we have recently been able to find is that a vast majority of the neurodegenerative diseases that we see come about due to oxidative stress leading to inflammation and inflammation leading to expression of the neurodegenerative diseases based off of do we have the actual genetic predisposition for those neurodegenerative diseases. Within these different types of neurodegenerative diseases, we can have things that can impact the cells themselves, such as with Alzheimer's disease, the ability to propagate signals, such as what we see with Parkinson's disease or with multiple sclerosis, or with Huntington's disease, or with amyotrophic lateral sclerosis, ALS. What ends up happening is we end up having this kind of vicious cycle taking place, where we end up having our healthy neurons that somehow have some sort of damage to them. That damage leads to a misfolded protein. That misfolded protein leads to aggregation of misfolded proteins, which leads to filament aggregation around the neurons, which leads to fibril formation around the neurons, which leads to excessive damage, which ends up impacting the healthy brain or the healthy spinal cord or the healthy neurons or the healthy nervous system within the body, leading to additional issues within the nervous system. So what are some of these neurodegenerative diseases? Well, we have things like multiple sclerosis. We have things like Huntington's disease. We have things like Alzheimer's disease. We have things like Parkinson's disease, in which we have different types of fundamental root causes for the diseases to come into play. Where with Alzheimer's disease or with post-concussive issues or chronic traumatic encephalopathy, we end up seeing plaque formations coming into play due to inflammation. With Huntington's disease, we end up losing neural tissue due to genetic predisposition to having neural tissue degenerate. With multiple sclerosis, we end up losing myelin. Whereas with Parkinson's disease, we end up losing the ability to produce neurotransmitters. Within all of these different types of diseases, what we end up seeing is we end up seeing distinct types of protein aggregations, what is usually referred to as plaque formations in and around the neurons. Some of them are referred to as amyloid plaques, where we can have diffuse plaques or dense plaques that we see within the nervous tissue themselves, where we end up seeing very small areas being impacted, and those small areas being impacted cause large cascading events to take place within the tissues themselves. These amyloid plaques can lead to loss of neural tissue. That amyloid plaque can lead to the degenerations that we see in things like Alzheimer's disease, can lead to loss of neural tissue, such as what we see in some cases as relates to chronic traumatic encephalopathy. We can also have other types of proteins, which are referred to as tau proteins. A lot of central nervous system neurodegenerative diseases are sometimes referred to as a tauopathy, a tau-opathy. That is where we see tau proteins intertwined within normal proteins in and around the axons or in and around the dendrites or in and around the synapses of all of the neurons that are being affected. Tau is an extracellular protein that is going to impact the ability for the axon to function in a correct fashion. Sometimes this tau can lead to tangles in and around the cell body itself of the neuron, which is going to limit the ability for the propagation of the action potential by not allowing the axon hillock, the area within the neuron that is going to allow for the action potential to be generated, to reach action potential. We also will see sometimes these misfolded proteins take place within the cell itself as opposed to outside the cell itself. So amyloid and tau we usually see outside, whereas with some of these other types of filamental formations, such as with the Lewy body or the Lewy nucleates, or with the within some of the glial cells where we see cytoplastic protein aggregation taking place. What ends up happening is we end up getting specific areas where we get exclusion of tissue, where the tissue gets replaced by these abnormal protein structures within the cell, abnormal cytoskeletal structures or abnormal neural skeletal structures, since we're dealing with neurons here, that impacts the ability for the neuron to function correctly. What this does is this leads to an inability to correctly propagate signals, inability to correctly respond to the neural signals taking place, inability to integrate signals at the various points within the neuron as we go through. There are other areas where we might see taus taking place, where we see taus in and around the cell body themselves, or unfortunately within the astrocytes themselves. When we start seeing tau proteins taking place in and around the cell bodies, this is usually due to glial interactions on the cell body themselves, where we end up having interference on the dendrites and interference on the transmission of the signal from the synapse on the dendrite to the axon of the neuron. This is where we have to remember that signals being propagated are going to go from the axon terminal to the dendrite to the axon terminal to the dendrite through the pathway. And what ends up happening is that we end up having interference on that pathway propagation based off of this tau accumulation at the dendrites or at the junction points of the dendrites with the supporting astrocytes. We start seeing distinct issues with the astrocytes in terms of these tau accumulations. What we end up having is metabolic interference with the neurons. And this metabolic interference with the neurons means that we do not get an appropriate neural environment for the neuron. That is, the environment of the nervous system is not appropriate for that neuron to remain functional. And one of the things that takes place when we start looking at that idea about the neurons and its propagation and its growth of the axons and its branching of the axons to form new synapses, or the synaptic pruning that takes place, is that these cells are secretory cells. And if they stop secreting, they start to undergo their apoptotic cycles. And when they start undergoing their apoptotic cycles, that's when we start to lose more neurons than what is there. And that is the hallmark of most of the neurodegenerative diseases, is that we're losing neurons faster than what we can regrow the neurons that are there. And so it doesn't matter in terms of what we're looking at in terms of those protein accretions, whether it's a tau, whether it's an amyloid, whether it's the TDP proteins that we see here in terms of the inclusion or exclusionary proteins, that is, are the inside the cell or the outside the cell. What they're doing is that they're creating an inhospitable environment for that neuron to do the neuron's function, which means it's not secreting correctly, which means it's going to start to undergo its apoptotic cycle, or we're trapping too much inflammation in and around the neuron. And this is where that inflammation in and around the neuron leads to excessive amounts of tau and amyloid on top of the neuron, and the excessive amount of tau and amyloid on top of the neuron leads to excessive inflammation within the neuron, and that excessive inflammation within the neuron leads to, via a vicious positive feedback loop, additional tau and amyloid protein, which causes the loss of the neuron itself, which leads to the reduction in what we see in terms of particularly the central nervous system neurodegenerative diseases, where we actually start seeing wasting away of neural tissue. We actually start seeing a reduction in the folding of the cerebral cortex of the brain. We actually start seeing little divots, little pockets of openness within the cerebral cortex itself. Most of these neurodegenerative diseases are going to be characterized by misfolding of physiological proteins. These are proteins that are normally occurring, but because of inflammation, because of other things taking place, they misfold. And when they misfold, they cause more misfolding to take place. The root causes that we see for these are associated with either trauma or oxidative stress, or we can have a genetic predisposition for them. The traumatic issues with these neurodegenerative diseases is what we see with head trauma, with concussions, with chronic traumatic encephalopathy, leading to issues within the limbic system, issues within the prefrontal cortex that lead to all of the behavioral modifications, all the behavioral issues that can come about following head trauma. Oxidative stress issues is what we're starting to notice in terms of root cause for a lot of our metabolic diseases that are lifestyle, put quotes around that, metabolic diseases that lead to neurodegenerative diseases, which is why we started to think about Alzheimer's and Lewy body and other inflammatory linked neurodegenerative diseases to be associated with metabolic diseases like diabetes. And it's one of the reasons why some people started referencing Alzheimer's disease as a type 3 diabetes. And that's because we start having, with oxidative stress, poor response to growth signals. We can have genetic predisposition for having these neurodegenerative diseases, such as what we see with multiple sclerosis, such as what we see with Huntington's disease. But we also see modification of certain genes associated with certain metabolism properties for individuals who are predisposed to expressing high amounts of oxidative stress, and by expressing high amounts of oxidative stress, being predisposed to having neurodegenerative diseases. There is considerable overlap within clinical phenotypes of the neuropathology and of neural loss, which means that it's very difficult to indicate what is specifically taking place within a lot of the neurodegenerative diseases without doing definitive diagnosing. And examination of the brain is the gold standard for the diagnosing. This makes it very difficult to have a definitive diagnosing on a neurodegenerative diseases, which is why a lot of times it's a suspected issue or it's a like issue when we start having these diagnosing of neurodegenerations. One of the ways that we've been trying to look at, particularly with Alzheimer's disease, because Alzheimer's disease has been on the rise within the population, is doing some sort of brain scan to identify Alzheimer's. But the problem is that just because we're looking at a specific protein, which is the amyloid protein within Alzheimer's disease, the amount of amyloid that we see being stained doesn't necessarily correlate with the person expressing Alzheimer's, which makes it very difficult to have a good gold standard diagnosing for things like Alzheimer's without doing a postmortem examination of the brain. This is why, for a lot of people, there's been some speculation that we've been pursuing the wrong avenue. And that's because what we're doing is we're looking at symptoms and not root causes as what we're doing for diagnosing. Very similar to what we do in terms of diabetic conditions, where we are looking at the symptom of hyperglycemia, high blood glucose, as being the root cause for the disease, as opposed to being the symptom of the disease. Just like having amyloid plaque is a symptom of having Alzheimer's, but it may not be the root cause for the Alzheimer's, which have led to recently a lot of investigation into ion deficits and antioxidant deficits as being one of the root causes for increased amounts of oxidative stress and expression of Alzheimer's, where we've also started to notice changes in distinct types of metabolism taking place. And the distinct types of metabolism taking place leading to increased amounts of inflammation within astrocytes that can be part of the root cause for things like Alzheimer's disease. Well, thanks for listening. Hopefully thanks for watching. Hopefully got a bit out of the discussion about neurodegenerative diseases. We will talk more specific about specific neurodegenerative diseases in subsequent talks. Please make sure that you are liking and subscribing to what we're putting out there. Give us a big thumbs up if you are enjoying what's there. Please make sure you've subscribed. Please follow us here on YouTube as well as on the podcast, the written stuff in Substack, as well as the quick takes on Instagram and Threads.